First, I just want to say thanks everyone for coming, and I feel really honored to speak in front of all of you. I'm going to be talking a little bit about some solutions, more solutions that we have for breeding, especially for resistance and also productivity, and how can we leverage big data and global partnerships to accelerate genetic gain in wheat and get these improved varieties out to farmers faster. It's been said that in the next 40 years, humans will need to produce more food than they did in the previous 10,000. I think this really underlines the problem and the challenge that we face. We know that the current rate of productivity gain is not sufficient. So here are global projections of productivity gain over time for staple food crops. And this dotted line shows what we need in comparison to what is projected. And so if we continue at the current rate, it's just simply not enough. So we need to ramp up this rate of improvement. We talked a lot about rust and disease resistance, and what we really want at the end of the day is productivity. And so we need to protect the yield, but also productivity is a function of many traits, including the intrinsic grain yield, also protection against many other diseases, not only stem rust, but many others pictured here. And also keeping in mind that the product must be acceptable to the consumers. Now the problem is even more complex in that all these different traits which underpin productivity express themselves differently in different environments. And different environments require a different suite of traits. So, a line that performs really well in a cool, well-watered, high-input environment is not going to be the same variety that performs well in a hot and dry. So really makes the problem even more complex. Part of the solution to solving this problem is breeding. So what we mean by breeding is just this continual cycle, what starts by crossing two parents and then generating progeny, evaluating those progeny, and then doing selection. And that process continues year after year after year, and mankind has been doing this for thousands of years, but of course in the past 50 or 100, we've been able to really advance the science of breeding and do things better and better. And what we're trying to achieve with breeding is genetic gain. And what I mean by genetic gain is the improvement in performance of in this case, wheat, due to genetics. Luckily, we know how to increase the rates of genetic gain. So we know that genetic gain per unit of time is based on four variables. One is the selection intensity, so how strict are we at selecting, and we can increase this by increasing our population sizes. Another is the diversity, which you need to have something from which to select, and then selection accuracy. So how can we best identify really truly superior lines? And then the last one, which is also one of the most important factors, is the time. So I mentioned that cyclical process. The faster we can turn that crank and the faster we can do those cycles, the greater the rate we're going to be able to achieve. So we want to reduce the time. Luckily, in wheat breeding, we have these wonderful public goods to support genetic gain globally. And one of these is the centralized breeding effort and global phenotyping network led by the CGIAR research program on wheat, especially headed by the institutions CIMMYT and ICARTA. So I'll mention the CIMMYT breeding program a little bit later on. And this global breeding effort has really a broad reach, and that breeding operations happen on a large scale at this institution, and then data and germplasm flows to partners, and then with that feedback we can really make progress. We already heard about the phenotyping platform in Kenya, so the International Stem Rust Screening Nursery is another excellent public good, and we hope to have more of these kinds of screening nurseries to really enable these genetic gains in wheat. We also have some new technologies which at our disposal, which are really exciting and promising. And one is genomic selection, which using whole genome marker data and prediction models for selection of the best individuals. And what I mean specifically by genomic selection, like how does it work? We take genotypes and phenotypes on a population of wheat lines. We use that to train a prediction model, and then that prediction model is used to predict a set of selection candidates, and we can then select on them even before we see them in the field. And so this enables us to turn that crank faster and to more quickly identify promising lines. We know genomic selection works. This is information from a world-leading animal genetics company. So genomic selection got its start in animal breeding, and they've been implementing this now for a while. And so this company this is a pig breeding program. They significantly accelerated their rate of genetic gain after introduction of genomic selection. You can see on this graph the dotted line that's happening is around 2013. This when they started implementing, and you can see that trend line of progress really ramped up after they implemented this technology. And genomic prediction models work on a relatively simple premise that information is borrowed from relatives. So if we know that the more closely related two individuals are, the more they're going to resemble each other. So by genotyping on a genome-wide scale, we know the precise relationship between two individuals, and so we know that information for all individuals in the population, how their relationships are. And so then we can borrow information from close relatives in order to predict other members of the population. And so what a genomic selection integrated wheat breeding program might look like, now it's not too different from an ordinary breeding procedure where you would intermate the parents, generate candidates, and then at that point, as soon as you have the candidates, you can predict them and then start to make crosses again. You also then, you advance some of those candidates through advanced testing, so we still do the phenotyping and the dissemination. And even at those points in time, we can also make predictions for dissemination. So which lines will be better adapted to which environments. And then the information from testing and after lines have been disseminated feeds back into the prediction model. So we have this cyclical model training process and the cyclical breeding process happening at the same time. We applied genomic selection for stem rust resistance breeding. This began in 2010, part of the Durable Rust Resistance in Wheat project. And our target was breeding for quantitative resistance for better durability. So with stem rust resistance and with some other disease resistances, we have different kinds of resistance, some based on many genes, so the quantitative type, and that is shown to be more durable. So we target, and genomic prediction works especially well for these quantitative traits. So we implemented this. One also want to point out another advantage to using genomic selection, especially when we're talking about quarantine diseases. We have this resource of screening nurseries, but when you're talking about a quarantine disease, you're limited by the physical space that you cannot bring the pathogen to any location in the world. And there's a physical limit to how much phenotyping you can do. So you can imagine the scenario where you can phenotype maybe thousands, maybe hundreds of thousands of individuals in a disease nursery, but then you can predict another 100,000 or even millions based on genotype only. So the scale, the genotyping scales up a little bit easier than you can with phenotyping, especially in a quarantine disease situation. So our main objective was just to implement genomic selection for stem rust resistance and measure the gain from selection. We started off with training our model with data from Kenya and Ethiopia on a population from CIMMYT, and applying this model in CIMMYT germplasm. And our genotyping was just genome-wide genotyping markers linked across the genome. And so these are the results that we got. We actually were able to see realized gain, and we did this in, we did two replicates of the selection experiment, and in both we saw gain from genomic selection. So here, just showing you the histograms, and the dark blue is the populations which we started with before selection, and the light blue is the populations after 2 cycles of genomic selection. And we were able to shift the mean closer to 0, so we were able to get more resistance on average in these selected populations. So we were successful. And genomic selection, of course, it doesn't discriminate on traits. It works for really any trait, especially is suited for quantitative traits, but most of the traits that we're working with are quantitative. And so this is just give you an idea of some of the work, some of the research that has been done showing that genomic selection works for other traits in wheat. There's also another advantage that we can take advantage of with genomic selection is we can accelerate genetic gain for multiple environments. So often in breeding, we have this problem where we have our breeding environments where we're testing, and they may not be the exact same environments where we'd like the varieties to be grown. So for instance, we might breed in one location, but then share the germplasm with another site, which is not the exact same environment with which we did the selection. And so it's a kind of indirect selection which we're doing. Now what we can do to try to improve that is actually use data from these sites where the lines will be grown and use that in our prediction model to help us improve our indirect selection. And so just to give you an example of what we're doing, we're testing out this approach with a project to use genomic selection to accelerate genetic gain for heat tolerance in South Asia. So this is a project led by Kansas State University with collaborators at CIMMYT and Cornell. And we have these 4 sites in the wheat growing areas of South Asia. These are sites that are prone to heat, so heat is one of the primary target. Heat tolerance is a primary target. But we also have to keep in mind all the other important traits that are important for farmers. And we cannot forget about UG99 resistance as we do this. So we know that UG99 resistance will be important for South Asia because it's in the migratory path of the pathogen. And so we need varieties with both resistance and high yield under heat stress and non-heat stress, in addition to many other traits. But just to give you an idea of the partners that are and the players that are involved and who's doing what and what are our roles. So we have the UG99 screening nursery pointed out here because we use information to ensure that the lines are resistant to UG99. Then we have the yield and especially under heat stress, also data from other traits coming in from South Asia. We have multiple traits being evaluated and the core breeding operations happening at CIMMYT Mexico, also prediction modeling happening at CIMMYT Mexico, and then methodology research and genotyping happening at Kansas State University and Cornell University. And then we all have to, the data has to flow across these collaborators and in order to drive this forward. So this is kind of the approach that's being taken. So there's phenotyping that happens in Kenya and Mexico. So after the candidates are developed, they're then phenotyped. Then we predict their multi-environment performance. So we predict their performance, especially in South Asia. So we have a phenotype from Mexico and Kenya. We predict performance in South Asia. Then we send the best individuals for South Asia. We then receive data back from those sites and then use it to update the model again, so we can repeat that process. Under the conventional way, we would still do this, but we wouldn't have that added information from the prediction model. We would just have the phenotype from CIMMYT in Mexico and then send the best individuals based on that information alone. And we have some indication that this is actually helping us to improve selection. So in here I'm just showing you on the blue bar is showing the indirect conventional selection. So just using the information from Mexico, how much, what's our selection accuracy if we want to improve the yield in these sites in South Asia. Then in the orange we're showing if we integrate all the data, so if we use all the data, including data from Mexico and the data from South Asia, in addition to the genome-wide marker data for the prediction modeling, we're able to improve our selection accuracy above what we would get with the conventional method. So this gives us an indication that this approach is actually allowing us to make greater genetic gain. So next, I'll just briefly talk about high-throughput phenotyping. It's another really exciting technology which is coming on, and this is just using proximal or remote sensing for rapid and automated phenotyping. And so this is a picture, a little bit of what high-throughput phenotyping looks like. So this is an unmanned aerial vehicle doing remote sensing over some wheat plots, and what this takes thousands of images and stitch them together, and then information can be extracted. So here's an example just of what some of the images look like coming off of these vehicles. So on the left is a visual image, and so thousands of these are taken and stitched together, and a lot of data can be extracted. And on the right is a thermal image, and we can extract temperature from this kind of imagery. We've been able to show that it's possible to improve our genomic prediction accuracy by integrating this high-throughput phenotypic data. So here's an example from a heat-tolerant or heat-stressed environment for grain yield. And on the right, you can see ordinary genomic prediction accuracy. On the left is a genomic prediction accuracy where we have used the data collected from remote sensing on our candidates to help us improve our ability to predict. And you can see that when we have really good reliability of data, we can achieve this greater accuracy. And so how this would fit into a breeding program is it's similar to what I showed before with the genomic prediction integrated breeding, except now in our prediction model, we're integrating not only the genome-wide marker, conventional phenotyping, but also high-throughput phenotyping to make our predictions just more accurate. So we have a lot of challenges. Of course, these technologies are really great, but there's a lot of hurdles. So one of the most important ones and the most challenging ones that we face is efficient data input and output. So we don't have yet good databases for the raw data coming from sequencing and image data. And so this is really challenging because we can't use our conventional relational databases anymore. We also need pipelines to extract information from the raw data. So in sequence data, we want genotype calls and then a set of quality markers. From image data, we want plot level measurements and then phenotypes. And so we still are in the process of developing these robust pipelines. Another one is knowledge gaps. I mean, with the fast pace of the changing technology, it becomes really difficult to stay updated, and there's a huge demand for learning opportunities. We just hosted a short, breeding technology workshop, and there was tons of interest, hundreds of applicants. We could only take 40, and we really only scratched the surface of these technologies, and it's just a lot of knowledge sharing that needs to happen. Of course, we're not going to be able to do all of this unless we work across multiple disciplines. And so we really need to get out of our silos and start working across discipline. And this can be sometimes painful, but it's really, really important because a lot of the solutions have already been figured out by in these other disciplines. Also, I think in the future, we're really moving towards having multiple experts contributing to breeding. So I talked about the multidisciplinary work. And it used to be that the breeder really makes most of the decisions with input from others, but now we're kind of moving towards a system where you have multiple people contributing to the decision making, and the decisions are becoming more and more objective and data driven. And so really all these people all these experts are the breeding unit, and instead of it being just one person really making the decisions. Of course, we still need somebody in the driver's seat, to make everything go in the right direction. But we're really moving towards this more objective, more data-driven approach. And I think these couple of images really underline the transformation which is happening. So on the left, we have breeders in a field looking at weak spikes, including Norman Borlaug, and on the right, we have some breeders in the field looking at aerial imagery that was just collected from a UAV on a laptop in the field. Of course, I don't want to say that, I don't want you to leave with the impression that breeding is going to go away, or we're going to be replacing breeding with high-tech solutions, because breeding, the activities, vast majority of them will remain the same. So we still have to do the crossing, we still have to do culling selection, we have to do phenotyping, the yield trials, the harvesting, the threshing, and it's still, there's still a lot of steps we can't avoid. So we're still going to do breeding, but the way we do it is going to be different in a sense that the decision making will be improved, and we might have to tweak how we do the processes to take advantage of our technologies, but we still need to do all these. So just to wrap up, I hope I've convinced you that big data, global partnerships can help us increase rates of genetic gain for all key traits, and this is key for delivering productive and disease-resistant varieties to farmers. And ultimately, breeding decisions will be objective and driven by data, enabled by international and interdisciplinary breeding teams. And technologies will enhance breeding, but they're not going to replace breeding. And that, I'd just like to acknowledge our sources of funding, Bill and Melinda Gates Foundation and DFID from the UK, supporting through the Durable Rust Resistance in Wheat project. Also, I mentioned the project in South Asia is funded by USAID Feed the Future project. And the CGIAR also supports operations at CIMMYT.